Okay, anatomy people, this video now will focus on the clotting factors, the platelets. But again, I hope at this point you're now experts on your erythrocytes and your leukocytes. Right, so let's get now into the platelets. Okay, so when we begin platelets, we begin with a very familiar cell. And again, I've been really focusing on this cell. So again, very, very, very important for the exams. Very, very important beyond that for just your knowledge on how these formed elements work. So the hemocytoblast. So the cell, cyto, that gives rise, that produces blood, formed elements of the blood. So in this developmental pathway, we see some things happening. So we go to a megakaryoblast, and then we see some changes, and then we see this. So we see some fragmentation here, and we see some fragmentation of the actual cytoplasm with little kind of like granules in there. So, but not granulocytic, it's not a white blood cell, right? So when we talk about platelets, platelets themselves are not actually even cells. They're just going to be little crumbs, little fragments of cytoplasmic, little cytoplasmic extensions here, cytoplasmic fragments. So platelets, also known as thrombocytes. I don't like that word because they're not technically cells. They're not sites. They're just little fragments of a cell, of a megakaryocyte. So platelets are not cells. Platelets are cytoplasmic fragments of very large cells called megakaryocytes. A big nucleus cell. So it's a cell with a big nucleus that's fragmenting apart. So these platelets are going to function in the hemostatic process, in the process where they stop bleeding. This is a very important aspect, right? So if there's any damage, trauma to the blood vessels, we need to be able to go in there and seal that up so we don't have massive loss of blood. So hemostasis, the stopping or stoppage of bleeding. Platelets have granules, but these are different granules than we see in the leukocytes. The leukocytic granules are for defense. The platelet granules are for clotting, for coagulation aspects. So hemostasis, I'll show you a little video that kind of gets into more detail, but there's a couple of things that happen at the blood vessel level. So we distinguish, this is at the cell level. There's things that happen at the actual tissue at the blood vessel level. Right? So one, we're going to have vascular spasm, platelet plug formation, and then finally coagulation. So somebody got a little scratch there. So three things would happen. So vascular spasm, so the smooth muscle, which you remember from last semester, right? 2401. So the smooth muscle senses some sort of injury, so that it tenses up, right? It constricts, and by doing so, it narrows then the diameter of that vessel. So by doing that, we're going to slow, like a traffic jam. We call it traffic jam. It's going to slow the process of flow through there. So any type of injury, certain chemicals that will occur, pain receptors, all of that can cause that spasm, this contraction of smooth muscle within the vessel wall. So the spasm. Two, we then start to work and form that plug. These are the platelets. So it's a series of reactions that are going to happen. It's a reaction, it's a cascade reaction that happens to make our actual clot. And then we're going to end up with this fibrin mesh that coagulates everything here. So this is just an overview. We're going to get into some specifics now. So when we talk about a blood clot, what is actually a blood clot, right? It's a network of thread-like protein fibers. So it's a network of fibrin. Fibrin is this sort of like spiderweb looking protein that will trap blood cells, platelets, and fluid. So in order to get to fibrin, in order to form fibrin in the vessel, we're going to go through a series of reactions, and it's going to be triggered by a series of coagulation factors and also calcium. So clotting pathways can be quite intricate. I'm going to introduce them. I'm not going to necessarily get involved with each individual chemical reaction, but we're just going to kind of look at it in the big picture idea here, right? So there's two paths to get to the final product, kind of like in math. There's multiple ways to solve problems, but they get us to the correct answer. So here we're going to have an extrinsic clotting pathway and an intrinsic clotting pathway. Two different paths to get us to the same final fibrin mesh there. So both are usually triggered simultaneously during a tissue damaging event. And it's kind of like a domino effect. So one little molecule triggers the formation of another, and that one triggers in the formation of another and another. And it's just kind of this feedback mechanism that leads to that final end product of fibrin. Okay, so here we get into some detail that, again, is going to be a lot of stuff, and I'm not going to necessarily ask this level of detail for this first exam, but when we say clotting factors, well, the factors actually have names, right? Fibrinogen, prothrombin, tissue factor, calcium, procelerin, so each one of these has an actual name. Depending on who you work for, if you're working in a phlebotomy aspect, a lot of people just memorize them as factor 1, factor 2, factor 3, factor 4, factor 5. So these are the factor numbers and the actual factor name. Now, what are these factors? They're basically, most of the time, they are proteins. It's a plasma protein, plasma protein, plasma protein, plasma protein. So these are proteins that just kind of hang out. They're constantly in circulation within the plasma. Right? Some of these are ions. You know about calcium. It's not a protein. It's just an inorganic ion. But most of them are proteins, and most of them are naturally produced by the liver. Liver. So it shows us an importance of, well, yeah, the liver detoxifies a lot of things we put in our body, but the liver actually is an important source for clotting factors. So if somebody ends up with hepatitis, somebody ends up with cirrhosis of the liver, we're not producing the proper amounts of these plasma proteins, which are clotting factors. You can see how it would have an impact, a negative impact on blood clotting itself. So right here, this tissue factor, it's a little bit different. It's a glycoprotein tissue factor that's actually from the cells, from the tissue, from the cells themselves, not from an organ. It's the individual cells themselves that release this tissue factor. So to put everything in one messy slide here, right? So intrinsic pathway, right? We start with intrinsic pathway, vessel endothelium ruptures exposing underlying tissues. So collagen. So collagen becomes available, becomes accessible to the plasma itself and triggers this reaction here. So this factor leads to this, leads to this, leads to this, leads to this. Eventually, we're going to end up all the way down through the pathway to form prothrombin activator, right? So here's factor 8 that helps with this process. We'll talk about factor 8 in a bit. Extrinsic, tissue cell trauma exposes blood to tissue factor. So the actual cells that contribute this substance, this clotting factor, the glycoprotein. So with the help of calcium, right? So it's a little quicker, a little bit faster reaction, still gets us at the same place. So it's a lot of individual steps and details, but the moral of the story, we get to the same prothrombin activator. That's important, right? So intrinsic and extrinsic still arrive at prothrombin activator. It's important. sort of a junction for both of these pathways. So let's look at again, here on the right, the extrinsic. So here we have a blood vessel that's been ruptured, right? So this extrinsic pathway is initiated by thromboplastin or tissue factor 3, which are chemicals extrinsic to the blood, right? So there's substances on the outside that don't normally come into contact with the blood inside. So when that vessel ruptures, now we have these damaged tissues, damaged cells that release the tissue factor 3. The intrinsic pathway is initiated by chemicals inside of the blood responding to exposed collagen fibers in damaged blood vessel. So it seems kind of similar. Let me see if I can make an analogy here. Imagine in your house, so you have the wall, right? Your sheetrock wall. If we were to tear that sheetrock wall, behind we have that insulation, that pink fiberglass insulation maybe. That irritates you. It causes irritation, makes you itchy. But you're not normally exposed to that because it's behind that wall. So for us to be exposed to the fiberglass insulation, we have to damage the wall. Once it damage the wall, then we can expose ourselves to that fiberglass. So we don't normally get exposed to collagen inside of the blood vessel, but if there's trauma to that blood vessel, on the inside of the blood vessel, there's collagen fibers in that that cause this reaction, right? So again, from the vessels themselves, basically. I hope that made sense. So regardless, if it's an extrinsic, the tissue factors, or intrinsic, the collagen, and normally it's both at the same time that are working, we get to that prothrombin activator. There's two pathways to prothrombin activator. Once we get to prothrombin activator, then we go to a singular path, right? So it's a common pathway to thrombin, and then thrombin is going to initiate the formation of fibrin, our final blood clotting pathway. And that's what fibrin mesh looks like a big spider web. So there's our erythrocytes that are getting trapped and stuck on the actual fibrin mesh. So this is an important part of our clotting mechanism. Again, more sticky fibrin, sticky fibrin, sticky fibrin, sticking the erythrocytes here. So if you want to try this at home, get the sticky duct tape, sticky face out, and just put all this crazy duct tape all over the doorway, and then call your little brother, little sister, see if they can kind of easily come in, right? They're going to get all stuck with that sticky duct tape. It's kind of like the idea here. So we have a rupture. We have a hole in the blood vessel. Red blood cells are trying to leak out. Well, this fibrin mesh is going to trap them, so then they're not able to leak out. So if you ever had a cut, you see it was bleeding, a little cut, eventually it stops bleeding, but then there's still some of this yellow fluid that oozes out for a little while. Well, plasma can still leak out for a bit here. That's what that fluid exudate is sometimes called, right? So that's where that yellow fluid can still leak out for a bit until that becomes completely sealed. So what are we talking about here? The prothrombin activator, right? Extrinsic leading to prothrombin activator, intrinsic leading to prothrombin activator, which then helps to stimulate the formation of thrombin. Thrombin initiates or activates with fibrinogen to produce fibrin, which is our actual clotting mechanism there. Now, again, so all part of that, those three steps to we have the spasm, we have then the fibrin mesh, and then finally the coagulation of all that. Now, once all of that is done, we have to go in there and remove the clot. If that's not removed, then we have other complications here, right? So we have some of these hemostatic, these clotting disorders, right? Thrombembolic conditions. So one, we have a thrombus. we should not have a thrombus. If everything is working correctly, we form the clot in the blood vessel, but then the clot has to get sort of destroyed, digested, so then it allows natural circulation through that area. If we have a thrombus, we have a clot that develops and persists in unbroken blood vessel. So two things can happen. One, we were supposed to form the clot because there was an injury. The clot formed, but the clot was not removed. Or two, which is different and potentially a little bit worse, there was no damage, but yet we started a clotting cascade incorrectly and formed a clot in a non-broken vessel, right? So that's a thrombus. Bad. Right? Worse is when we have an embolus. This is when a thrombus or an embolism is where the thrombus then breaks, detaches from the wall, and then travels, circulates in the body. So why is this bad? Because not all vessels are the same diameter. There's some thick vessels with big diameter lumens. There's some really tiny capillaries. Well, if this free-floating blood clot is circulating through all of these little tiny vessels, normally it will clog up a vessel and damage that the area distal to that clot. So this is then the basis for strokes, the basis for clogged situations and not allowing blood flow into these areas. So not a good thing here. So these are basically a clot, or we're not supposed to have a clot. Down here we have then these bleeding disorders where we're not clotting enough. We're not clotting when we need a clot. So the two opposite situations. We've heard the term penia before, right? A deficiency. Now we have the idea of thrombocytopenia. So thrombocytopenia, not enough blood clotting. So for whatever reason, we don't have enough platelets, or the platelets don't have enough of the clotting factors. For whatever reason, we're not being effective in the clotting cascades here. Right? We mentioned if there's potential damage to the liver, that can affect our overall number of clotting factors. If there's genetic conditions, right? Hemophilia, let me come back here. In hemophilia is a genetic condition where a child doesn't produce factor VIII. So it makes it very inefficient to go from here to this pathway down to prothrombin activator there. Right? So it's a genetic clotting deficiency there. So hemophilias, impaired liver function, thrombocytopenias, all kind of related in the ideas that we're not having enough clotting, right? So not enough platelets, not enough clotting factors, or genetic abnormalities in the clotting factors, or genetic deficiencies in the clotting factors. So something that I work with, right? I work with a lot of venomous reptiles and looking at some of the effects of the venom. So this is a case study. I'll post an associated video with this to show how the venom actually does what it does, right? So if I'm a hungry snake and I bite a rat, I want that rat to die so I can easily ingest it, right? So I could get injured. The rat can bite and scratch. So I want to inject this venom, let the venom do its job so I can have lunch. Well, what does the venom actually do? In this case, for this particular species, this venom targets this sort of clotting cascade. And I'll let you watch the video and you can see how, what effect the venom has on the actual blood. Right? Oh, okay, this next bit I don't want to get into now. It's going to take a while to get through that. So I'm going to stop the video here. And tomorrow I will continue with the transfusion reactions, right? And it'll be the last part for this first chapter. But this gives you a lot of material to kind of go through today and tomorrow. And again, I'll have that video up for you tomorrow with the transfusion reactions. But for now, this is a good stopping point. So you all have a good evening, and I will be seeing you again tomorrow.